Hello, students. In today's session, let us learn about mitosis. Here, you can see the animation of how a cell splits into two daughter cells. And both the cells are similar in its composition, gene composition, and cell content. Introduction. In cell biology, mitosis is a part of the cell cycle in which replicated chromosomes are separated into two nuclei. Cell division gives rise to genetically identical cells in which the total number of chromosomes is maintained. Therefore, mitosis is also known as equational division. In general, mitosis is preceded by the S stage of interphase, during which the DNA is replicated, and is often followed by telophase and cytokinesis, which divides the cytoplasm, organelles, and cell membrane of one cell into two new cells containing a roughly equal share of these cellular components. The different stages of mitosis altogether define the mitotic phase of an animal cell cycle, the division of the mother cell into two daughter cells genetically identical to each other. The process of mitosis is divided into stages corresponding to the completion of one set of activities and the start of the next. These stages are prophase, prometaphase, metaphase, anaphase, and telophase. During mitosis, the chromosomes which have already duplicated condense and attach to the spindle fibers that pull one copy of each chromosome to the opposite sides of the cell. The result is two genetically identical daughter nuclei. The rest of the cell may then continue to divide by cytokinesis to produce two daughter cells. The different phases of mitosis can be visualized in real time using live cell imaging, producing 3 or more daughter cells instead of normal 2, which is actually a mitotic error called the tripolar mitosis or multipolar mitosis. Other errors during mitosis can induce apoptosis, which we've already learned, or it can even cause mutation. Certain type of cancer can arise from such mutations. Mitosis occurs only in eukaryotic cells. Prokaryotic cells which lack a nucleus divide by different process known as binary fission. Mitosis varies between organisms. Animal cells undergo open mitosis where the nuclear envelope breaks and then the chromosomes separate. Whereas in case of fungi, the mitosis is a closed type where chromosomes divide with an intact cell nucleus. Most animal cells undergo a shape change known as mitotic cell rounding to adapt a near spherical morphology at the start of mitosis. Most human cells are produced by mitotic cell division. Important exceptions include the gametes, that is the sperm and egg cells, which are produced by meiosis. So mitosis is applicable only to the somatic cells of the body, whereas the reproductive cells, they divide via meiosis. Then how was this discovered? Various attempts regarding cell division were made in the 18th and 19th centuries. In 1835, the German botanist named Hugo von Moll described cell division in green algae, Cladophora, stating that the multiplication of cells occurs through cell division. In 1838, Matthias Jakob Schleiden, he affirmed that the formation of new cells in their interior was a general law for cell multiplication in plants, a view which was later rejected in favor of Moll model due to the contributions of Robert Remarque and others. In animal cells, cell division with mitosis was discovered in frog, rabbit, and cat cornea cells in 1873 and described for the first time by Polish histologist named Wacław Mazel in 1875. Buchley, Schneider, and Paul might have also claimed the discovery of the process, which we refer as mitosis. That is in 1873, German zoologist by name Otto Buchley. He published data from observation which he made on nematodes. A few years later, he discovered and described mitosis based on those observations. So this term mitosis was coined by Walter Fleming in 1882, which is derived from Greek word where mitos means warp thread. That is something that is used to warp around. These are some alternative names for the process. It is also known as karyokinesis, which was actually introduced by Schleicher in 1878, or it is also known as equational division, which was proposed by Mr. August Weismann in 1887. However, the term mitosis is also used in a broad sense by some other authors to refer to karyokinesis and cytokinesis together. Presently, equational division is more commonly used to refer to meiosis II, the part of meiosis which is almost like mitosis. Here is the summary, the image which summarizes the different phases of mitosis. So here you can see the interphase stage where the DNA is seen in the form of a chromonema, that is like a thread, and the centrioles are formed. In the prophase, you can see how the centrioles move and they form the early mitotic spindle. In the pro-metaphase, the mitotic spindles are clearly formed, and the chromosomes, the condensation of the chromatin fibers into chromosome is seen. In metaphase, they all arrange near the equatorial plate, and in the anaphase, the sister chromatids are pulled to the opposite poles, and in telophase, the sister chromatids are separated, and in cytokinesis, the cell divides, that is, the cytoplasmic content of the cell divides between the two cells. Prophase. This is the first phase in mitosis. During prophase, which occurs after G2 interphase, the cell prepares to divide by tightly condensing its chromosomes and initiating mitotic spindle formation. So during interphase, the genetic material in the nucleus consists of loosely packed chromatin. But at the onset of prophase, chromatin fibers, they condense into discrete chromosomes that are typically visible at high magnification through a light microscope. So in this stage, chromosomes are long, they are thin, and also thread-like. Each chromosome has two chromatids. The two chromatids are joined at the centromere region. So here you can see the microscopic view of prophase that happens during mitosis. Gene transcription ceases during prophase and does not resume until late anaphase to early G1 phase. That is the gap 1 phase. The nucleolus also disappears during early prophase. Close to the nucleus of animal cells are structures known as centrosomes. They consist of a pair of centrioles surrounded by loose collection of proteins. The centrosome is the coordinating center for the cell's microtubules. A cell inherits a single centrosome at cell division, which is duplicated by the cell before a new round of mitosis begins, giving a pair of centrosomes. The two centrosomes polymerize tubulin. The two centrosomes, they polymerize tubulin to help form a microtubule spindle apparatus. Motor proteins then push the centrosomes along these microtubules to opposite sides of the cell. Although centrosomes help organize microtubule assembly, they are not essential for the formation of the spindle apparatus since they are absent from plants and are not absolutely required for animal cell mitosis. The next stage is prometaphase. At the beginning of prometaphase in animal cells, phosphorylation of nuclear lamin causes the nuclear envelope to disintegrate into small membrane vesicles. As this happens, microtubules, they invade the nuclear space that is available, and this is what is known as open mitosis, and it occurs in some multicellular organisms. Fungi and some protists such as algae, trichomonas, undergo a variation called as closed mitosis, where the spindle forms inside the nucleus and or the microtubules, they penetrate the intact nuclear envelope. In the late prometaphase, kinetochore microtubules, they begin to search and attach to chromosomal kinetochore. A kinetochore is a proteinaceous microtubule binding structure that forms on the chromosomal centromere during the late prophase stage. A number of polar microtubules find and interact with the corresponding polar microtubules from the opposite centrosome to form the mitotic spindle. Although the kinetochore structure and function are not fully understood, it is known that it contains some form of molecular motor. When a microtubule connects with the kinetochore, the motor activates using energy from ATP to crawl up the tube towards the originating centrosome. This motor activity, coupled with polymerization and depolymerization of microtubules, provides the pulling force necessary to later separate the chromosomes to chromatids. Next, moving on to metaphase. Once the microtubules have located and attached to the kinetochores in prometaphase stage, the two centrosomes, they now begin pulling the chromosome towards the opposite end of the cell. The resulting tension causes the chromosomes to align along the metaphase plate or the equatorial plane, which is an imaginary line that is centrally located between the two centrosomes, almost midline of the cell. To ensure equitable distribution of chromosomes at the end of mitosis, the metaphase checkpoint guarantees that the kinetochores are properly attached to the mitotic spindle and that the chromosomes are aligned along the metaphase plane. If the cell successfully passes through the metaphase checkpoint, it proceeds now to anaphase. Here in the image, you can see that roughly the chromosomes have arranged themselves in the center of the cell through an imaginary line, which we call it as a metaphase plate. The next state is anaphase. During anaphase, the cohesion that binds sister chromatids together are cleaved, forming two identical daughter chromosomes. Shortening of the kinetochore microtubules pulls the newly formed daughter chromosome to opposite ends of the cell. Polar microtubules push against each other, causing the cell to elongate. In late anaphase, chromosomes also reach their overall maximal condensation level to help chromosome segregation and the reformation of the nucleus. So here is another microscopic view of the anaphase. See, you can see that the sister chromatids have been pulled towards their opposite poles. Next is telophase. Telos means end. Basically, it is a reversal of prophase and prometaphase events. At telophase, the polar microtubules, they continue to lengthen and elongate the cell even more. If the nuclear envelope has broken down, a new nuclear envelope forms using the membrane vesicles of the parent cell's old nuclear envelope. The new envelope forms around each set of separated daughter chromosome, though the membrane does not enclose the centrosome, and the nucleolus reappears. Both sets of chromosomes, now surrounded by new nuclear membrane, now they begin to relax and decondense. So the mitosis is complete. Each daughter nucleus has an identical set of chromosomes. Cell division may or may not occur at this time depending on the organism. Here you can see within a single cell, two sets of chromosomes are formed. So this indicates the telophase stage. Followed by telos phase is the cytokinesis, that is a prophase, metaphase, anaphase, and telophase. They constitute karyokinesis, that is the nuclear division, whereas cytokinesis is the division of the cytoplasmic content. So it is always the karyokinesis that is followed by cytokinesis. Cytokinesis is not a phase of mitosis, but rather a separate process which is necessary for completing the cell division. In animal cells, a cleavage furrow or a pinch, which contains a contractile ring, develops where the metaphase plate used to be. So pinching of the separated nuclei. In both animal and plant cells, cell division is also driven by vesicles, which are derived from the Golgi apparatus, which move along the microtubules to the middle of the cell. In plants, this structure coalesces into a cell plate at the center of the phragmoplast, and they develop into cell wall separating the two nuclei. So the phragmoplast is a microtubule structure which is very typical for higher plants, where some green algae uses phycoplast microtubule array during cytokinesis. So each daughter cell have a complete copy of the genome of its parent cell. So the end of cytokinesis marks the end of mitotic phase of cell division. Here in the image, you can see the cytokinesis. So there's a contractile ring creating cleavage furrow, and it therefore creates two different cells with in each with a set of chromosomes. In case of animal cells, it's a furrow that is formed. In case of plant cells, a plate is formed, cleavage plate, which further divides the cell into two. There are many cells where mitosis and cytokinesis occur separately, forming single cells with multiple nuclei. The most notable occurrence of this is among the fungi, slime molds, and cenocytic algae, but the phenomenon is found in other organisms also. So even in animals, cytokinesis and mitosis may occur independently. For instance, certain stages of fruit fly embryonic development, we can see that they occur differently. Cytokinesis and mitosis, they occur differently. Yes, moving on to the function of mitosis. Mitosis function or significance relies on the maintenance of the chromosomal set. So each formed cell receives chromosomes that are alike in composition and are equally numbered to the chromosome of the parent cell. Mitosis occurs in the following circumstances: development and growth. The number of cells within an organism increases by mitosis. So this is the basis for development of a multicellular body from a single cell, that is zygote, and also the basis of the growth of a multicellular body. Cell replacement. In some parts of the body, that is skin and digestive tract, cells are constantly slowed off and replaced by new ones. New cells are formed by mitosis and are exact copies of the cells that are being replaced. In a like manner, RBC cells have a very short lifespan. They have about only 4 months, and new RBCs have to be formed by the process of mitosis. Mitosis also helps in regeneration. So some organisms can regenerate body parts. So the production of new cells in such circumstances is achieved by mitosis. Say, for example, starfish. If an arm of a starfish is lost, it can regenerate with the help of mitosis. Also, mitosis serves as a means of asexual reproduction. So some organisms, they produce genetically similar offspring through asexual reproduction. For example, the hydra, it reproduces asexually with the help of budding. So the cells at the surface of the hydra, they undergo mitosis and forms a mass called as bud. So mitosis continues in these cells of the bud, and this grows into a new individual. And the same division happens during vegetative propagation in plants. So to summarize, the primary result of mitosis and cytokinesis is the transfer of parent cell genome into two daughter cells. So when the mitosis begins, the chromosomes, they condense and they become visible. The nuclear envelope, which segregates the DNA from the cytoplasm, now disintegrates into smaller vesicles. The nucleolus, which makes the ribosome in the cell, now disappears. Microtubules, they project from the opposite end of the cell and attach to the centromeres and align the chromosomes centrally within the cell. The microtubules then contract and they pull the sister chromatids of each chromosome apart. Sister chromatids at this point are now known as daughter chromosomes. As the cell elongates, the corresponding daughter chromosomes are pulled towards opposite end of the cell and they condense maximally in the late anaphase stage. A new nuclear envelope forms around the separated daughter chromosomes, which now decondense to form interphase nuclei. So during this mitotic progression, typically after the anaphase onset, the cell may now undergo cytokinesis. In animal cells, a cell membrane pinches inward between the two developing nuclei to produce two new cells. In plant cells, a cell plate forms between the two nuclei. Thank you.